In this short video, we're going to take a look at gram staining. Now, the materials you're going to need are clean microscope slides. They may have to be cleaned with a methanol pad before we use them. We're going to need our gram stains, and we should get everything ready before we're going. Of course, we need a specimen and something to apply the specimen with, lots of paper towels and distilled water. Now, you can either use a colony from a Petri plate, or you can use a colony from broth, as shown here. And the best way to transfer these to the slide is to use a sterile swab. And this is the proper technique for peeling out back the paper cover. And you'll notice the technician is holding the holding the tube in his left hand, and is holding the cap in the right hand using the pinky finger, and is twisting it back and forth with the left hand. We're very careful, of course, not to touch anything with the swab until we do our transfer. Now, this is showing a microscope slide on a template, and the template, which is printed on paper, has spaces for A and B. We might compare a gram positive and a gram negative strain, or maybe we're comparing an unknown rather, to a known strain. So there you can see the two applications. Next, it goes to the slide warmer, and typically that's 5 to 10 minutes, but you need to check and make sure it's dry. If it's not dry, leave it on the slide warmer for a longer period of time. Now, this is one example of how we might treat the slides. You can see the clothespin there, which is going to be used as an inexpensive slide holder. This particular rack, which is in the sinks in T231, is very good because we can flood and clean up fairly easily. Now we're applying the gram crystal violet dye, and that's going to be for 30 seconds. So we're going to put 2 to 4 drops on each spot. So we have an A and a B spot, so we're going to have then 2 spots of the crystal violet stain. And then we'll count for 30 seconds. And then we need to wash the slide off. And so we're going to hold it with the clothespin, and then using distilled water, we're going to wash it very vigorously. And as you recall, the crystal violet is going to be taken up by both cells. You can see he actually washed both the back and the front of the slide. Now we've returned it, and we're going to add the grams iodine, which we're going to leave on for 60 seconds. Now this is going to form the CVI crystals, the crystal violet iodine crystals. Now iodine is a mordant. It's going to complex with the crystal violet. Crystal violet is soluble, but the crystal violet iodine crystals are not. So you can see the two areas, which we could actually see visually blue. We're not going to let the whole 60 seconds elapse here in this particular slide. Now, we've washed them, and we're decolorizing. Now, we flood the entire slide with decolorant, which is 95% ethanol acetone. And really, you just leave it on for a few seconds. It's not a very long procedure. And then we want to, again, rinse very quickly with water. Now, the acetone alcohol decolorant is going to be, will remove any unreacted crystal violet. So any crystal violet iodine crystals will remain behind. And then finally, we want to use a counter stain, which is safranin, which is left on for 60 seconds. And again, safranin stains everything. So gram-negative cells, which were decolorized, will now be stained with the safranin. Again, washing the back of the slide and washing the front of the slide. Now we need to blot. And so we're going to place the slide face up onto the blotting paper. We're going to fold it over one time. And then we're going to press down. Do not rub the slide. That's very important. If you rub, you're going to rub off the specimen, which is loosely bound to the surface, and you'll have tear and streak marks. We're just going to push down and hold it down. And that's it. The procedure's done.